So my name is Elias Koutis.
My name is Elias Koutis, and unfortunately Bence Kendernoy is too sick to attend, so we'll be taking over his part.
Okay, so this is our project on molecular biology, and it all basically started when my grandfather started noticing these strange markings on his peach trees. Now, since he makes his living from his peach trees, we had to find what was causing it, and we believed it to be the PPV, which is a widespread virus that can be found almost anywhere in the world, but only two types are present in Europe, the PPV-D and PPV-M. Now, it's not harmful to humans, but it's not that nice to consume. It causes chlorotic rings, as you can see, on the petals, leaves, and the fruit as well. And it causes a loss of production and yield. So what we did is there's different areas of his peach plantations, and we took one area and took 5 samples from that area. Okay, so we tested those with 2 different methods to make sure that the PPV was actually the cause.
Okay, so first of all we used a biochemical process known as the PCR, which stands for polymerase chain reaction. And we use this method because it is excellent for the detection and diagnosis of infectious diseases, which in our case was the Plumppox virus. So basically we gathered the samples from my grandfather's field, and we extracted 100 milligrams of them and put them inside a mortar. And along with some extraction buffer and some sand, we grinded them. And then we placed these homogenates into separate Eppendorf tubes, as you can see here. But before we could amplify our products, we had to first purify them, which took us about 3-4 hours to complete. Basically, we kept them inside water baths at specific temperatures and centrifuged them a couple of times in order to separate the large unwanted molecules from the rest. So once here you can see before purification and after. And once we were done with the purification, we had to use reverse transcription as our so far we were working with RNA viruses, and the PCR machine can only function with DNA. So once this was finished, we went on to amplification, and after amplification we Bence has made us a gel plate into which we placed our samples along with a DNA marker. We put it inside the electrophoresis apparatus and ran electricity through it. After about half an hour, we took the gel plate out and placed it inside the UV transilluminator, which was connected to the computer. And from this we gathered our results. And from here you can see that we tested first for both the PPV-D and the PPV-M virus strand. Here we tested for the PPV-D and here for the PPV-M. So here you can see our 5 samples. These are 3 positive controls, and this is 1 negative control, as it shows negative in all 3 separate testings for the virus. Now you can see here that 4 samples show positive for the presence of either PPV-D or M, but sample number 5 shows an unusual spectrum. So we came to the conclusion that our 5th sample got contaminated during the preparation process for this virus testing. Now, here you can see when we tested separately for the PPV-D virus, that sample number 2 and 4 show positive, and in the case of PPV-M, the sample number 1 and 3 show positive. But sample number 5 shows negative in both cases. Therefore, we came to the conclusion that 4 out of 5 samples were infected by the PPV-D, and only 1 sample, the 5th one, was free from viruses.
To make sure that our results were correct, we did a second experiment called ELISA, which stands for Enzyme-Linked Immunosorbent Assay. Now, the main difference between the 2 experiments is that ELISA concentrates on the protein body within the virus, while PCR concentrates on its genetical material. So first we started with extraction, which meant that we placed our samples into plastic buckets, and using this high volume squeezing machine, broke up the cells into organelles, which contain the protein body. Then using microlayer plates, which were designed especially for this experiment, since scientists every day have to work with a lot of samples. Fortunately, we only had 5. First, we placed an enzyme-linked antibody into the microdurl plates, which is also called an immunoglobulin. And then we placed our extract in it as well, and then another layer of immunoglobulin. And last but not least, we added a substrate which indicated the presence of the protein body by a color change. And by doing this experiment, we were now sure that it was the PPV that was affecting the peach trees.
Okay, so basically our grandfather's field is a 10-acre land, and it's made up of 5 2-acre plots. And we tested a 2-acre plot. You can see here, this is the 2 acres. And what we did is we took 5 different samples from different parts of the field and tested them separately for the presence of the virus. This area here is the one where the virus symptoms were most visible, and this is the only part of this plot where the virus has not been present yet. So thanks to our investigation, we warned our grandfather in time to spray the rest of his field with pesticides, but sadly the rest had to be cut down. So thank you very much for listening.